I've got the gardeners in the back, so I'm going to have to move. Give me a second. Meanwhile, for everybody, it's my pleasure to introduce Bhramar Mukherjee and her postdoc, Max. Sorry, I've forgotten your last name.
Aung.
Aung, yes. Who's from the University of Michigan. Bhramar gave a superb talk last week on a paper that I had seen in manuscript a while ago, which did such a fine job of addressing the problem of high-dimensional mediation, a problem that I had been thinking about then at that point, and she so totally scooped the idea that I had started to develop and did such a much better job of it that I decided to abandon the problem entirely. So I'm very excited for her to share her work with us now. She is, if you're not already familiar with her, the chair of Biostat at University of Michigan, and well known to us for her contributions in both genetics and environment and gene-environment interactions and a whole host of other things, interests that overlap so closely with our work. So with that, enough said. I'm going to mute myself so you don't hear the gardener blowers in the background. And why don't you go ahead? You want to share a screen? Have I made you, I guess I need to make you a host first.
Yeah, so you
Make co-host. And I guess you're joining separately. So Max, I'm making you a co-host as well.
Great, thank you.
I think you should be ready to go. Are you able to share yet?
yes. So I'm just trying to do one thing, and I'll be ready in a minute. So thanks, as we
I'll just tell you about your audience, which are faculty, staff, students, postdocs in our Biostat division primarily, but we often have people joining us from cancer epidemiology, environmental health, and other divisions as well, or even other departments.
Yeah, so I can see that I can share my screen, but before I do that, so first of all, thank you, Duncan, for the nice introduction and mentioning that I tackled the problem that you have been thinking about. I have always noticed that there is this uncanny similarity in terms of what we get interested in. Sometimes you scoop me and sometimes I scoop you, but in the end, the world wins. But I also want to say that there is a reason behind this similarity, because I always follow your footsteps. So you are someone that I really admire. I read your papers and I said, oh, Duncan is thinking about it. So maybe this is something that we can all work on. You have been a true role model for me. I just want to present this talk as a tribute to your work. So I actually pasted
and vice versa, I should point out.
So I pasted a link in the chat function. So it is, I find it weird to give a talk in webinars because you don't know your audience. So what I thought that I'll do is it's basically like sending out a message out there in the world and in the universe, and somebody is intercepting. It's that feeling. So I thought that I'll start with some polls to try to really learn about my audience. And I will you will see the data, and as soon as you start voting and clicking on it, and you can see the screen, right? That the results of the poll. So I just wanted to know, like, how are you guys feeling about webinars? And some people think this is very good because you can see the slides and you have an intimacy, but some people it's just hard. I can tell you that this I'm asking you as an audience, as a speaker, I think it's really, webinars are really, really hard. So I just want to see how you feel about it in this group. All right. So it seems like that we do not like webinars as much as in-person seminars, probably. I don't know whether the difference is statistically significant or not, but so for the next 1 hour, you'll have to bear with the situation that we're just having to deal with because of our new circumstances in our lives. So the second question I want to ask, because we have been hearing so much about the pandemic and the negative effects on it, on our health, on our society, on politics. And so I just want to ask, but we have also adapted as human beings. We are very resilient. We have also adapted. So what is a new skill you have learned during the pandemic? So if you just type in, then I should be able to see the results. Can you see the screen? Yes. Working from bed. Python. You can see the results, right? Max, can you see the results on my screen?
Yes.
All right. Thanks. Running, sketching, present in webinars, yes. Making ice cream, early morning walks. All right. So the last question is that I think as educators and scholars, we are really thinking about this very hard. And this is just for my own interest, that we are really thinking about what will happen when the pandemic is actually ending. We are learning every day about online presentations, online webinars, online education. What is going to happen to us? What is the biggest challenge facing higher education in the post-pandemic world? Hard opinions. Jobs, yes. In-person social skill. Harder to justify expensive tuition, yes. I am afraid to talk in person in face now. So that's an interesting one. I'm not quite there yet, but I think I'm getting there. All right, so thank you for participating in the poll. And I'm now going to go back to business and the rest of the talk. So Duncan asked me to talk about some of our work on Bayesian methods for high dimensional mediation analysis. And as I said, that this is a series of work and jointly with many collaborators in genetic epidemiology as well as environmental epidemiology at University of Michigan, because this analytical problem and challenge actually appears in both domains. So my slides look clear? Okay.
Yep.
Okay. All right. So first, I would like to talk a little bit about mediation analysis. So obviously, mediation analysis is a very old topic, almost 100 years old now. And what is the new twist? And why do we still need to work on mediation analysis for particularly high dimensional data and high dimensional mediators? And then I'm going to talk a little bit about the Bayesian mixture model work that we have been doing for high dimensional mediation analysis. And again, there are many people in this department who have contributed so greatly to the literature, and we all learn from our collective work and make progress in research. And then the final thing I'd like to really talk about is something relatively new, that when you do variable selection in a regression model or a high dimensional regression model, then we always think about penalization of the regression coefficients. And we can think about many methods like lasso, elastic net, your very favorite spike and slab type Bayesian variable selection, or continuous shrinkage priors. But when you are doing mediation, you probably, your target objective function that you want to optimize has probably changed. You really want to pick up those mediators with natural indirect effects. So how to target selection of mediators and change your penalization algorithm is something that I'm going to spend a little bit of time on. And then I'm going to conclude my part, and Max is going to take over and walk you through a very exciting case study with application to environmental epidemiology. And I must say that Duncan said a lot of things about me, but my research is really always standing in the shoulder of my mentees and my trainees. And it was wonderful to have Max in our lab, and you'll see that what he has accomplished. So I want to talk with this, start with a little bit about history of mediation analysis. I just mentioned to you that the first mention of mediation analysis in a historical context we see in the work of Sewall Green Wright. He was a geneticist, and a famous paper by him is also the first paper where we talk here about path analysis and also correlation and causation. These are old papers, and as I was reading these papers, I was thinking that if any of my papers is going to survive 100 years, so that's a wonderful feeling that people are still reading this work. So he proposed the idea of a mediation analysis in terms of a structural model where the effect of an exposure or a treatment or an intervention is mediated to certain processes, biological processes or social processes. And we try to model that, try to capture that, and to make some assumptions. And you'll see that mediation analysis is wrinkled with many assumptions that we are going to make about exposure mediator, outcome mediator, all of these relationships. So as soon as this new idea was proposed, this was 1920, 1921, there was sweeping condemnation of the idea. And this is a quote by Niles, another geneticist at that time. And he says that this type of methods will be a great value to science, but if only it were valid. And it is certainly disappointing that you are putting together a simple graphic system which truly represent, is it ever possible to really verify the assumptions that you're making? And some criticisms actually really stand the test of time. If you think about causal mediation analysis and when the natural direct effect or natural indirect effect has a causal interpretation, we layer our models with assumptions which are often not verifiable by data. And so that was the same criticisms in Niles, that you can postulate what you want, you can say any kind of DAG, and it would be beautiful in terms of logic and science, but it is really, is it realistic or is it verifiable from the data that we have? And so this all started with this path analysis and mathematics of path coefficients. And again, if you see the potential outcome theory and the counterfactual framework, this is some of the common criticisms that have come up, that it is pure mathematics, but it is a lot of algebraic manipulation because you're saying you're changing your intervention, you're changing your mediator value, but really it's very far devoid of any reality. So then Wright came back with this criticism, the response, the rejoinder to the criticism, a rebuttal to the criticism, and you can see that it is so detailed. So I feel like, we have, how does academia actually progress? And so this was a great example of that people try to question what you have done and you defend. And by defending that, by refining that idea, you actually improve yourself and improve your science. So I feel like, in a sense, it is very good to be a little feisty as a profession and get involved in arguments and vibrant debates. So you can see that, again, this is a very long rejoinder where he tries to talk about, defend this idea of mediation analysis. So coming back to the history, 100 years first, fast forward, we are still talking about mediation analysis. And many of you are quite familiar with this basic, mediation diagram, where you have an exposure. And throughout this talk, I'm going to denote that by A, or an actionable variable. And then you have exposure mediator relationship. And then you have mediator denoted by M and outcome denoted by Y. So there is a direct effect of A on Y, which is we are going to denote by beta of A. And then there is an indirect path of effect of A, which is going through this mediator. And typically, depending on which product model or difference model you think about, it is some kind of product and a function of this alpha A and beta M. So in our example today, we are going to think about exposure not as a binary treatment variable, which is very popular and the commonly known context in causal inference, or a multiple categorical treatment variable, but actually a continuous random variable, which is the environmental toxicants. And M, this is the place where high dimensionality is going to come up. You can think about A could also be high dimensional, and we can talk about that situation too, but mostly it's the high dimensional mediator, and agnostically putting in a bunch of biomarkers that you have measured. It could be an epigenetic biomarker, it could be endogenous biomarkers, which Max is going to talk about these measurements, and then Y for our case is going to be several birth outcomes. And we have looked at premature birth, we have looked at preterm birth, and we have looked at gestational age, head circumference at birth, birth weight, various metrics of childbirth. Now, we look at the testing question. So typically, when you are interested in identifying the mediator, that whether there is any kind of indirect effect through this path or not, or it is all the direct effect, the total effect is a decomposition of the direct and the indirect effect, then you are interested in testing this hypothesis. If you were actually interested in testing as opposed to estimating the indirect effect, the null hypothesis is product of two things are 0 versus product of two things are non-zero. This leads to a very interesting statistical question. The question is now that the null hypothesis, product of two things are 0, that means we have to get back to in how many ways can a product be 0? And so the first case is both are 0, and the other two cases as one of them is 0 and the other is not. So the null hypothesis now becomes a composite. Yeah, go ahead. Is there a question or?
No, I accidentally hit the power. Sorry.
No worries. And again, please feel free to stop me, because this is a very informal and intimate discussion. We all want to understand what's going on here. And so, and then you have the second case that, you have the null hypothesis as a composite hypothesis. So this is unusual in statistics, because we always think about the alternative as a composite hypothesis. Our null tend to be simple. So you can decompose the null into 3 compartments. And as you see, as I would build more complex models, this is really the key, that there are different compartments of mediation or indirect effect structures that we are trying to capture. So one is where both are non-null, that's our signal. That's our true signal in terms of mediation effects. The others are thrown into the bucket of the null, but they also have some structure to them. So there are several new papers on single mediation analysis where people are thinking about how to optimally test when you're agnostically look at genome-wide mediation, for example, and there are extremely clever papers on how to really improve the classical test of mediation analysis. Particularly, I'd like to refer to the work by Huang in Academia Sinica. He has done some brilliant theory around this problem. So let us go back to then how people would normally test this hypothesis. So we know the classic Sobel test, where we take the product of a parameter and we replace by maximum likelihood estimate. We do delta theorem in order to find out the standard error. And that's how we take the ratio, we get a test statistic. And this is all good and fine, but you can see that there is a problem with the denominator in our case when alpha and beta are both equal to 0. Would anybody like to say what happens when alpha and beta becomes close to 0 to the denominator? It blows, like the students, you can immediately recognize the problem that there is actually blows up. The standard error becomes close to 0, the test statistic blows up. So then the asymptotic distribution of this test statistic is usually people use a roughly approximate normal distribution, but you cannot really use that when alpha and beta equal to 0. So why didn't people encounter this? This is almost obvious when you stare at this problem, that this standard error is not going to work when alpha hat and beta hat are coming to 0, or the true parameters are close to 0. So the reason is that in earlier days, people used to do mediation analysis in a much more targeted way. thinking that I'm only going to do mediation analysis if I see that one of the alpha or beta are departing from zero. There is some effect of the exposure on the mediator, some effect of the mediator of the exposure on the outcome. If none of them are true, then I'm not going to really do the mediator analysis. So here I have actually dropped the suffixes A and M from just to make things clearer. I'll come back to that notation in a minute. So people avoided that case because that's when they just say that we are going to do some basic checks, that when do I want to do mediation analysis, and this will be a part of the check. I do not want to do things. So when everything is done. So what has changed now? What has changed now is that we are throwing a whole microbiome or an epigenome as mediator, and we're doing agnostic mediation analysis as opposed to targeted mediation analysis. That's why the landscape of methods have changed with the science, and as such, our statistical considerations have to change with the science. So what happens if we actually try to use the Sobel test statistic with an asymptotic Z distribution? It turns out that it is way more conservative than what it is supposed to be. So in this figure, I have plotted the standard normal curve and the distribution of the Sobel statistic, and you can see that the empirical distribution is quite far from the standard normal, and you will have a very conservative type 1 error. When it was supposed to be 0.05, you get into the order of 0.0001. So this is conservative, but in the other cases, as I said, people did not think about this because most of the time we are either in case 2 or in case 3 will do these checks. And in those cases, the distributional assumptions, asymptotic assumptions holds up fairly robustly. But then we really need to think about, can we do better in terms of a more powerful method and not such a conservative type 1 error as well. So now, and again, I refer to you to the work by Huang and also some by Shihong Lin's group in order to do this one at a time mediation analysis, and what are some optimal procedures for that for agnostic genome-wide mediation analysis. I'm going to move a little bit now. So now I have talked about the history. I have talked about what is wrong with the history. And then I want to talk about not just a single one at a time mediator, but I'm going to toss in the entire microbiome here, or have this p-dimensional mediator where p is quite large. So typically when you do multiple mediator analysis, yes, go ahead, please.
Oh, a quick question.
Yes,
so if you go back to the distribution comparison,
yes,
under both being 0,
yes.
I remember you said that when both are 0, the statistics tends to blow up, but here it looks like it's being conservative.
So so what we have done here is this the empirical distribution of this of the Sobel's test. And again, so in some cases, if you look at the asymptotics, it is going to blow up. But what we have done is to just show you the approximation. In some cases, in the alpha hat and beta hat are very, very close to 0 here, and that's the asymptotic distribution. And the normal 0, 1 I have put as a reference distribution. Yeah, it's not always going to happen, right? You can still derive actually the distribution of you can still derive, if I use this is going to blow up, but there is a alpha hat, beta hat on the top. You can actually try to derive the distribution and empirically plot it as well. Just that the variance calculation, the normal 0, 1 does not work.
Okay, thanks.
Yeah.
I also have a question about this hypothesis. So I think traditionally for when we do the mediation analysis, we want to know first whether the X is related with Y, right? And then also whether the X is related with mediator. Right. And then we do the mediation analysis. Right. So if there's no association, for example, in alpha or beta, then traditional thoughts is like we don't do the mediation analysis because there's no main effects.
Absolutely.
So what's the rationale that you bring that in? Because I think in practice we can do a stage-wise analysis. Like we can select those markers have the main effects, then put into the mediation analysis. So what's the advantage to have this?
Yeah, that's a great question. So exactly as I mentioned, that people did not run into this problem and did not really look up the encounter this particular situation of alpha equal to 0 or the mixture problem, because they will do exactly as you said, the screening first. That I'm not going to do mediation analysis. It does not make sense if I don't have one of these effects departing from 0. So we are mostly in these cases where you are okay. So then the sort of the the composite null, you are essentially by screening, you are reducing it to a much simpler null. But there are problems with any kind of two-step methods. Of course, you can think about a two-step method where you'll screen first and then conditional on whether you have some non-null effects, you are going to proceed to do mediation analysis. You have to tweak that first step threshold, how you're going to determine it in order to ensure the, the type 1 error and the power properties of the entire procedure. And actually in the Huang papers, they do compare that. The advantage of this, the throwing everything in, is that the context have changed. When you have millions of tests that you're doing, maybe a screening test, if you want to do it all at once and you want to understand that I want to run the same test across the whole microbiome or the whole epigenome, and we know that a large number of cases are going to fall in this both null compartment, can I accommodate that situation in a one-step model? And so that's where this is coming in. That if I have lots of alphas and betas, then if I try to group them, can I put them in the both 0-0 compartment, and can I distinguish the both non-zero compartment from the rest?
Yeah, that's good. Okay, thank you.
Yeah. Great questions. Thank you for the clarification question. And with respect to the Sobel distribution, actually under the null, so if you look at this test statistic, it's really, people have been using the Z alpha times Z beta. It is related to that test statistic, that if you just tested for alpha separately and beta separately and took the product of the two normals, and there is a lot of theory how the distribution of product of two normals are going to look like, so you can do a better job than using just the standard normal. And so if two things are independent, you can see that this is going to look like a chi-square. But then when things are not, then you can actually derive the distribution of the product of normal. There's 1940s papers and 50s paper on this distributional theory as well in that case. And some of that actually is used in Huang's paper to come up with a proper test and asymptotic null distribution for the test statistic. Okay. So what I want to do is that I don't want to do that, this is perfectly sensible that you do a screening of the mediators and reduce that set and only focus on the ones which have non-null effects. This is exactly like, I'm doing some invariable selection literature. I'm doing some kind of thresholding in order to reduce my dimension of the mediator. I do not want to do that. I want to be able to create a one-step fully principled process quantifying uncertainty so that I can pick out the non-zero compartment from the other three compartments. And how can I do that? So that's the rest of the talk and the methods. So again, now my notation is going to change because I was just describing one mediator. Now I'm going to have alpha A sub Aj and beta sub Mj here. And we also have the direct effect, which is of exposure, which is now one-dimensional only at this point. And you're going to have beta A, one coefficient there. But there are wherever the mediator was involved, I'm going to introduce a suffix j, and j is going to run from 1 through p, potentially large. And so the Max is going to talk about this example where the exposures are also composite exposures, that you have to do a dimension reduction in the exposure space as well. And for our example, it's different kinds of environmental mixtures, phthalates, phenols, metals, PAHs, and then the markers are measuring different biological mechanisms. And then we relate it to some outcomes. So if I wanted to do a single pairwise mediation analysis, I have 38 exposure divided into 4 groups here. And you can see the number of markers in each of the groups. And then I have 63, in this case, it's not too high dimensional, but 63 potential mediators. And I do not know the ordering, the causal ordering of them. It's an unordered set of mediators. And then I had to do approximately 2,400 tests if I wanted to do pairwise and did not impose any kind of screening threshold step. So So now what I was, so it is not optimal when I like to argue that it is not optimal to apply univariate mediation analysis when you have such a situation where your exposures are correlated, but the mediators also have a correlation structure. So it's the same kind of argument where you are going to do a single SNP test or a gene-based test when you have a correlated set of predictors. Can you gain more? So, and of course, there is confounding due to co-mediators that really leads to bias that we want to avoid. Also, if we throw in all of the mediators together and a bunch of them are correlated depending on the location of the mediators and other consideration, then there can be variance inflation and estimation instability as well. So a principal mediation analysis should probably think the whole structure into account and jointly handle all of these components in one sort of like framework, overarching framework. So I'm going to gloss through a little bit about the causal part, because I do want to talk about the Bayesian modeling and the penalization part, because the causal framework is nothing new in our work. We basically borrow the van der Waals framework of causal mediation analysis, but extend it to multiple mediators. So again, we have a situation where you have potential confounders that we are going to introduce, which are going to be denoted by C. We're going to assume that there are Q confounders, and then we have the mediators. But now when we write mediation analysis in a causal notation, I have to have two counterfactual. So this is something which is going to make our causal notations a little bit more complex, because I'm going to look at Y as a function of the exposure A and the mediator vector M. So that would be the ith subject's counterfactual outcome under an exposure value of little a and mediator value of little m. And now I'm going to, whenever I change A, M is going to change. If you look at the third bullet point here, mediators are also a function of the exposure. So every time I change an intervention and exposure, I have to think about the counterfactual for the mediators as well. So there is a lot of counterfactuals floating around, reflecting us back to Niles's criticism that these models are wrinkled and laced with many assumptions that you cannot verify. And we'll come to that assumption, those assumptions in a minute. And so again, then we'll define the natural direct effect and the natural indirect effect in a causal notation. And in the natural direct effect, we move A star to A. So A star is the reference level of the exposure or the treatment. And we move from A star to A, and we keep the mediator at the value if it was at the reference level, A star. So basically this is measuring the effect of the exposure, the direct effect of the exposure, holding the mediator at that reference value of the exposure. And then the average natural indirect effect we define where we actually change the mediator as well. So if if the exposure moves from A star to A, what happens to the mediator? So if you compare these two formulas, you can see the M part is the different part. When I have changed A star to A, then what is the mediator is also changing. I'm not holding that fixed. So that's the intuitive difference when we characterize the effect of the exposure, which is through the mediator. Now I have counterfactuals, which involves a lot of random variables like high dimensional mediator random variable, and we have to make assumptions. And this is where the problem is that we'll make assumptions about no unmeasured exposure outcome, mediator outcome confounding, and exposure mediator confounding. Those are the three arrows that you saw before. And those are huge assumptions that we know all the confounders. But then we also make a fourth assumption, which is not so well known, that no mediator outcome confounding can be affected by the exposure. So this arrow is actually broken, and the exposure cannot have this effect on the relationship of the mediator outcome confounding. So under these four assumptions, you can show that the causal interpretable functions that I defined here can actually be estimated by fitting regression models with the observed data, because neither do I get to observe the counterfactual for Y, nor do I get to observe the counterfactual for M. So now we'll come back under those assumptions. We are more familiar turf that we can write things in terms of regression models. And of course, there are assumptions about the form of the model. So we all know that. I'm not going to get into that because I want to really talk about how do we do shrinkage for this natural indirect effects. So now we have under those assumptions, we can write down our outcome model. So two key models, outcome model conditional on the exposure and the mediator model as a function of the exposure. So these are going to be our two key regression models. And you might just say, if I was looking at this problem and I did not know much about mediation analysis, the first my intuition is going to be, let me just do a shrinkage. Let me just do a shrinkage on these parameters of the on the betas, beta m's here, which is high dimensional in equation 1, and let us do a shrinkage or some kind of selection of the exposure mediator coefficients that are in all of these multivariate regression model. There are bunches of alphas that you have for each mediator. Let us try to penalize them and see which one is non-zero in each model and put the two together. That will be my natural first instinct to this problem, and we'll see that how we can evolve on that thought, starting with that good notion. So what we can do is that under this modeling assumptions, we can show that what is the expression for the natural direct effect and the natural indirect effect. And it so turns out that the natural indirect effect is actually composite of the sum from j equal to 1 to p of the product terms. So here I'd like to pause and really mention one subtle point, that each of those products for each mediator and exposure does not have a causal interpretation, because everything is conditional on what else is in the model. Only the sum has a causal interpretation in terms of the natural indirect effect through that complete pathway. And then we can say which mediator contributed to that pathway, but we cannot really render or assign a causal interpretation to each of the pairs of the product terms. So now we can see that if we move from A star to A, this is the natural indirect effect expression, and we want to quantify this natural indirect effect and want to find out which J contributes most to this natural indirect effect. So that's what our goal is. Now, and as again, these alphas, and now we have established that the individual alphas and betas are our key focus parameters, and we really want to target the product of the two parameters. Any questions? Okay. So now the game I'm going to play is that there are two ideas. The first idea that I mentioned, that I have two high dimensional models, two regression. I have so much machinery in terms of shrinkage and selection in my repertoire as a statistician. Let me just apply that to the two models and then just collect which ones are stand non-zero in the two models and put that group together. And then the other idea, because I'm seeing this product showing up everywhere, maybe I can do a direct penalization on the product. I think that's the novel part of our work, is this direct penalization of the product, the shrinkage of the product term in two regression models. So the first thing I did, as I said, that we followed our first instinct in our first paper, where because we want to do everything in a Bayesian paradigm to propagate uncertainty, to really understand these compartments of null hypothesis that I mentioned of alpha, beta, 0 and non-zero. So I wanted to put a continuous shrinkage prior, and I want to have a continuous shrinkage prior on the betas and a continuous shrinkage prior on the alphas. And I am going to put the two normal distribution. It's a mixture of two normal distributions. And I'm going to put one with a large variance, which is the first part, and the one with the small variance, which is the normal 0 sigma square m naught, that the small part. And so it's going to be a null and an alternative, m1 and m naught standing for the null group and the alternative group. And so a fraction of them are going to fall in the large variance component, and those are going to be our signals. So basically, this is what I'm after. I just, I changed the color. Sometimes it's not visible, that these are the ones, the large variance components are my components of noteworthiness, and I want to pay attention to what follows. And then I'm going to assume that what is the, when I choose pi m and pi a, this is the fraction of non-null signals in each of the model, that's where the sparsity assumption comes in, that I'm going to choose priors on this if I know something about it. Otherwise, we have standard priors for this mixture type of problems, and we're going to use that. So, go ahead.
Bhramar, can you
Yes.
elaborate a little bit on the advantage of using this mixture of two normals approach rather than a pure spike and slab where the density is zero?
Yes, so I think that the main thing for me is actually the computation. So if you have mixture of two normals, then I think the computation and what you see in terms of the speed of the algorithm is much faster. But also, I think that if you think about spike and slab priors, is it really like, do we ever really have the point mass at zero? People can argue from a philosophical point of view as well. And I also think that this prior helps us with estimation. The continuous shrinkage priors are designed to do estimation in a nice way. And there are ways to choose. There are recent results that if you actually can choose the prior parameters, sigma square M1 and sigma square M0 in certain rates, so that you can guarantee very strong frequentist performance and properties as well. But mostly, I think that there is a sort of a discrete structure to the spike and slab. It's extremely good at doing the variable selection. But when it comes to prediction and estimation, this class of priors are more amenable to that.
Thank you.
So now I want to pick out, so I want to achieve a lot of things. I want to achieve the estimation of the midway, natural indirect effect through that sum. But I also in that sum, I want to pick out components which are going to be the large component. And as in any kind of mixture prior, which has a spike and slab type components, a large variance component or a small variance component, then I want to pick out that which one. So we typically, when we do MCMC, we keep track of which component you belong to. So in this case, I want to keep track of both the components together. That's the only trick, that I want to find out that where my indicator variables are falling into the component membership, that is both of them are one. That's when I want to think about including that mediator as a noteworthy mediator. So I'm putting the two spike and slab or two mixture components together to find out what falls into my H1 component. And you can see we can also tabulate how many mediators fell into each of the null compartments as well. So I want to mention that, sorry, can I just mute myself? There is a phone call. I just want to turn my phone off. The Democratic National Party keeps calling, though I have voted. So I want to now rank this so that my posterior inclusion probabilities are going to be for the jth mediator are going to be just when both of these indicators are one. So that's the trick in this model. But what we want to really think about is the second model that I want to really come up with. This was a very good pass at it, but this was just the, that's what you think immediately. But when you think a little bit deeper, probably you want to really penalize the product terms. So how do you penalize product terms, and what is the literature on penalization of product terms in a regression model? And in a frequentist framework, there was a paper, and I want to really motivate this idea of penalization of the product term through a lasso type algorithm, because we are very familiar with those types of algorithm, where we have the first objective function, and then I want to really I want to penalize the product term in a way that I want to add a penalty to this likelihood function. And I want to look at it this way, that I have two models. So the first part is the likelihood of the y given AM model. And the second part is the likelihood of the multivariate likelihood of the multiple mediators given A and confounders. Confounders are always there. And then I want to tag on to this likelihood a penalty function. And now this penalty function actually looks very much like lasso or adaptive lasso. And this is from the work of Yi Zhao, who is an assistant professor in Indiana right now. And you can see that there is a new term. It's a product of the two coefficient and the absolute value of that, which is added to the penalty function. And in order to make the whole optimization problem convex, you actually add these two terms, which looks like a L2 term, makes it look more like an adaptive lasso type function for the rest of it. So you have the lasso type penalty, you have the L2 penalty, and then you have the product term. So this was called as pathway lasso, and their examples were high dimensional images as potential mediators and the signals at multiple images, and they used this successfully. So we wanted to not do a penalization like that, but borrowing idea from that automatically tells you that instead of modeling them as two independent priors for alpha and beta, I need to go into the space of a joint formulation. And that's what we did, that we want to look at a multivariate normal, a joint Gaussian mixture prior on jointly on alpha and beta. So I'm going to play the same mixture game, but I want to have three components exactly reflected into a joint model, where I have the completely null model, and then I have the models where one of them is 0 and the other is not. And then I have the full model, which is the first component, where I have variances and covariances, and I actually let that's my non-null signal component. So we are going to do a Gaussian mixture model for this model. And then one of my colleagues, Jian Kang, who works a lot on imaging and does a lot of thresholding prior, said that, okay, why don't we also do the penalization in a slightly different way, so that we actually generate from a Gaussian mixture, but then we do actually a product type thresholding, that you are going to, the unthresholded effects are just multivariate normal, just the standard assumption, but we are filtering out. We are letting people in when both of the betas and alphas are larger than a particular threshold. And also, so you're basically controlling the magnitude separately, and also the product exceeds a certain threshold. And then the question is, how are you going to choose this threshold in an optimal way? What are the prior choices of that? And we deliberated a lot on this, but also this is a product threshold Gaussian prior, which is fairly new, I think, in the literature. It has been used in imaging literature quite often. And so we basically then, now we have, so if you think about the journey of my talk, I started with the classical perspective, then I talked about the single mediator analysis, and then tried to motivate why we need to get to a joint model. And in the joint model, I talked about the Gaussian mixture model, and also the two normal mixture priors, two separately separable in betas and the alphas. And that method we call BAMA, Bayesian analysis of mediation analysis. But then all the other methods we can think about is that just do separate lasso on the two models. That's bilasso. We can do the Bayesian version of that, take the two models separately and do a Bayesian lasso. We can do pathway lasso. And the other methods that you're going to hear from Max about, that I did not talk about, is actually how to do dimension reduction, right? We can take that whole mediator vector and look at first two principal components or some representation in lower dimension in order to reduce the problem as well, to characterize the global pathway effect. We are not going to get back to our original interpretation like we have in these models, but that's also certainly a very sensible alternative. And so we have this landscape of methods, and you're going to see a lot of that from Max's work. But I just want to show you that what kind of output typically we get from this study, that the life code study, and Max is going to talk about it. It's a preterm birth cohort study. And I already showed you the schematic of the environmental contaminants that you had and the endogenous biomarkers and the covariates. So here I'm just giving you listing the particular outcome, the particular C, the particular M, and the particular A. And at the end of the day, you look for this kind of plots where you are characterizing the global effect of a pathway and trying to understand which pathway is more important than others. And which of the, so these are the PIP, or the posterior inclusion probability of a mediator, where both of the effects, alpha and beta, had to be larger than 0 and in a meaningful way. And you can see that this is the posterior probability, and we identify 3 potential mediators in this particular context using the Gaussian mixture model approach as well as the product threshold Gaussian approach. And so the question is that what does it really give us? And I think this is a mediation analysis. When you get to this, there was a question that was posed that why are we not filtering out? Why are we doing this entire microbiome or epigenome-wide mediation analysis? Does it make any sense? It's a very pertinent question. By doing this type of things, we are actually trying to learn about biology. If you think about the mediation analysis goal, it was mostly from a causal inference point of view of that if I cannot intervene directly on the exposure, if I can intervene on the mediator, then I can still attenuate or modify some of the effect of the exposure. So that was the original one goal of mediation analysis, is to try to understand intervention. But here it's completely different. When we're doing agnostic mediation analysis, we're really trying to understand biology and mechanism, that which group of mediators stand out relative to others, which pathway is more important relative to others. And so this work, so the BAMA paper appeared in Biometrics last year, and then we also released our package BAMA. Please do use it and let us know your feedback. And then, we just had the paper, application paper that Max is going to talk about, accepted in Nature Communication. This showed me and us a very natural progression of how science and statistics can go and move farther hand in hand. The last paper that I talked about is still under review. It's about sparse mediation analysis with targeted penalization. So I really appreciate feedback on this work, because with your criticism and comments only, this work will get better. So as a summary, I think that we have proposed some Bayesian methods which have competitive performance, and we have compared with a lot of other methods that you'll hear more about. And I think that we actually are trying to understand mechanistic pathways, and what we're trying to do right now is how to account for correlation, explicitly account how to borrow a selection of mediators when they are correlated, and also extend this method to multiple exposure. Another thing which have come up in one of my genetics applications is that we only have summary data. So in a genetics context, if we have summary data from regression models, and maybe I just know the outcome exposure model, and now I'm trying to borrow information from that auxiliary model to inform my mediation analysis and make it more powerful, that last problem is extremely intriguing statistically and mathematically, and I think is very important as well. So with that, I think I'd like to thank you and then hand it over to Max. I am happy to take any methods questions that you may have.
Let's hold those questions to give Max a chance.
Sure.
And then we'll come back to that at the end.
Absolutely.
Okay. So I budgeted for 15 minutes. So I don't know if the Zoom is going to close automatically.
I think the Zoom will continue, but some people might have to drop out.
Okay.
I know there's a class at 1 that some of our students will be attending.
All right, do you see my main screen with the full?
Yes.
Okay, awesome. All right, well, thank you everyone for this invitation. I'm really excited about this opportunity to talk about our recent data application with this really rich environmental data set. So I just wanted to begin by giving a little bit of background on the type of research that we do. So based on biomonitoring efforts, we know that humans are exposed to complex mixtures of environmental contaminants. And our previous work has been focused on characterizing the impact that these mixtures have on maternal health and the implications for fetal development and children's health across the life course. And this slide here illustrates the toxicant classes that we have had the capacity to investigate so far. And this fraction of the exposome includes non-persistent organic pollutants that Bhramar had mentioned earlier, which are the phthalates, phenols, and parabens, as well as toxic and essential trace metals, and more air pollutant polycyclic aromatic hydrocarbons. Now altogether, even with just these 4 toxicant classes, there's growing evidence from mechanistic models of in vitro and animal studies that these toxicants can act through several different biological pathways. And some of these mechanisms include interacting with circulating immune cells and enzymatic cascades to increase inflammation or alter metabolism, and also interfere with oxidative stress. So altogether, there's a need to understand how these toxicants are acting through some of these potential mechanisms. And as Bhramar mentioned earlier, mediation analysis is a powerful causal inference tool that we've been able to use to disentangle some of these relationships. And so we developed our mediation framework using data from the Life Codes cohort, which is a really great longitudinal case control study of preterm birth. And participants were recruited at Brigham and Women's study, or Brigham Women's Hospital. And this was in Boston, Massachusetts. And participants attended up to 4 study visits throughout pregnancy. And from those biological samples, we were able to conduct exposure assessment and measure for endogenous biomarkers. And so I also wanted to point out that from this larger cohort, we've been limited to this exploratory subset of 161 participants. And we recognize that this is a relatively small sample size, but our goal is to build upon this framework and really evolve the method to incorporate also longitudinal mediation analysis as well. So this figure here that Bhramar introduced earlier, it illustrates our conceptual framework for mediation. And we have this on the left panel, the exposure matrix that includes 38 individual analytes. And then in the center, we have our mediator matrix. And then in this particular setting, the outcome variable that we are focused on is gestational age at delivery. And now something that's particularly unique about these icosanoid panels that we've taken really interest and advantage of is that they can cluster into additional subgroups. And so they can be organized as either parent fatty acid compounds, or they can be organized as secondary metabolites that are governed by distinct enzymatic pathways, which includes the cytochrome P450 pathway, the cyclooxygenases, or lipoxygenases. And so based on these measured exposures and mediators, as Bhramar mentioned earlier, the status quo approach for characterizing mediation pathways is to build single pairwise mediation models. And you can see here that that would yield, right, over 2,300 individual mediation models. And so before I dive right into the mediation results, I wanted to take a moment to show you some results from a separate study that we conducted that really motivated this question of multivariate mediation. And it kind of echoes to one of the questions that the audience members asked earlier about doing sort of like this initial screening. And so this heat map illustrates associations between individual toxicants on the x-axis and mediators on the y-axis. And each of these cells is a regression coefficient estimated from single pollutant regression models. And so the red cells represent positive associations, while the blue cells represent negative associations. And the intensity of the color here, in other words, darker cells, indicate those with smaller p-values. And you'll also notice that some of these cells have green borders around them, and that means they're robust to false discovery adjustment. So here from this heat map, we can see at a really broad level that there are unique patterns for each of the toxicant classes across the mediator groups. And this really got us excited about building the foundation for multivariate mediation analysis. And I'll also add that in a previous study, we also learned through different prediction algorithms that the lipoxygenase and cytochrome P450 pathway of icosanoids yielded the greatest predictive capacity for classifying preterm birth, which further made the case for conducting mediation analysis with these biomarkers and these exposure analytes. And so when we look in and zoom into these pathways, we can see and recognize these unique patterns, Particularly, the phthalates exhibit substantially higher proportion of positive associations. And toxicology studies have also found that phthalates can interfere with these enzymatic pathways, particularly the cytochrome P450s. So stemming from these findings, in the first approach of our mediation pipeline, we approached analyzing this pairwise mediation analysis. And so this figure here illustrates the main findings from using that status quo approach. The y-axis here represents each of the individual toxicants, and the x-axis represents the negative log 10 p-value of just the mediation signal. So not the direct or total effect, but just the mediation effect. And so we can see from these results, there's initial evidence of mediation signals. For example, with this highlighted mediation signal with one of the phthalates and the icosanoid metabolite from the cytochrome P450 pathway. And so this is supported by the exposure mediator associations that I showed you in the heat map earlier. However, a majority of these mediation signals do not have significant direct or total effects that are distinguishable from the null. Therefore, we do not have sufficient evidence of complete mediation pathways when looking at single toxicants and single mediators. And I'll also add that these findings are also biased due to the fact that exposures within the exposure matrix and mediators in the mediator matrix are highly correlated with each other. So the single models that we built are by nature not independent models of each other. And so to be more intentional and thoughtful with mediation analysis, in this particular study and data example, we tried to circumvent this issue of multicollinearity by proposing to reduce dimensionality of the exposure mediator matrices. And in this setting, our approach was to create single exposure variables for whole toxicant classes, and then single mediator variables for whole biological mediator groups. And I'll walk you through a couple of the methods that we use to do this. So for the mediator matrix dimension reduction, one of the methods that we found particularly helpful is called population value decomposition. And this method reduces the mediator matrix by constructing orthogonal directions of mediation, which is similar to principal components analysis. And these directions of mediation are vectors composed of coefficients for each mediator variable, which are estimated based on maximizing the likelihood of structural equation models that incorporates information from the exposure matrix, as well as the mediator matrix and the outcome variable. So that's what sets it apart from doing simple principal components. And so after estimating these coefficients, we can take those coefficients and split them into these literature-derived grouping structures. So in our case, it was these enzymatic pathways, but if you're looking at epigenetic data sets or metabolomic data sets, you can use some of the other literature-derived grouping structures, such as gene pathways or biological clustering groups for the metabolome. And so once we do this estimation, we can take each of these mediator groups and apply matrix multiplication. And so here, each individual has an observed measurement for each mediator, and we take the linear combination of our coefficients against those observations, and then condense those mediators such that every individual now has just one observation for that entire mediator group. Now, for the exposure dimension reduction, we use a method where we construct environmental risk score using elastic net. And as many of you may know, the elastic net is a shrinkage estimation method that utilizes tuning parameters to identify the most important predictors and shrink the less predictive exposures towards zero. And so from implementing elastic net, what we can then do is extract those coefficients and then use them as weights to construct the environmental risk scores. And similar to how we condense the mediator matrix, we apply here for the exposure some matrix multiplication for each individual's observed exposure measurements to estimate a single environmental risk score for each of those toxicant classes. And after reducing the dimensionality of the exposure and mediator matrices, we can then conduct a more biologically informed and intentional mediation analysis. And so in this slide here, I'm using a forest plot to illustrate the main findings from multivariate mediation analysis. And here I'll just begin with a fraction of the results where we have all of the mediators analyzed simultaneously as one whole group in relation to the phthalate risk score. And from these results, we see evidence of complete mediation where the total effect signifies that the phthalate risk score is associated with reduced gestational age at delivery independent of the mediators. And after adjusting for the mediators, we see that the second parameter shows there's evidence of mediation effect when evaluating all of the mediators simultaneously. But we want to explore which of the pathways are contributing the greatest to this mediation effect. And so when we look at all of the individual enzymatic pathways that I introduced to you earlier, what we learned is that the cytochrome P450 pathway was contributing the greatest to the mediation effect for the phthalate risk score. And so we can conclude that exposure to phthalates is associated with decrease in gestational age, and that this effect was largely driven by that cytochrome P450 pathway. Now, when we visualize the results for the other risk scores, the metals, the polycyclic aromatic hydrocarbons, and the phenols, we do not observe evidence of mediation by any of the individual mediator groups. And overall, the significance of this approach is that it advances our ability to estimate precise risk profiles. It reduces issues of collinearity across the different exposure analytes. And our findings in this setting highlight the cytochrome P450 pathway as a potential biological intervention point for phthalate toxicity during pregnancy. So I just wanted to make some concluding ideas. And one of them is that the large body of science that our group has really been passionate about and focusing on has shown ample evidence that environmental toxicants can act through a diverse array of biological pathways. And this really necessitates mixtures analysis as well as thinking of creative ways to apply multivariate mediation analysis. I'll also add that there are inherent problems, right, with analyzing individual mediation models. And one of the ways to circumvent this was dimension reduction. And it allows us to quantify whole group mediation effects rather than looking at single biomarkers, which in many epidemiology studies can have very sparse effects, given that they're just looking at single biomarkers. And finally, I'll also add that this analytical framework, we designed it so that it can be flexible for multiple different omics domains. And so I've been working with collaborators to think of ways to apply this with epigenetic arrays, and I'd be excited to also continue the discussion with some of you to think about ways that maybe this can be applied to other types of data sets as well. So with that, I just wanted to acknowledge the LifeCode study participants and the collaborators, as well as the funding sources that have made this research possible. So thank you for having both of us here today, and I'd be happy to work with Bhramar in taking questions.
Well, thanks, many thanks to both of you. We can direct questions to either one of you. Anyone?
Yeah, hi, Tom. Actually, a very fantastic work. I'm wondering, like, can you tell us a little bit more why you choose this population value, like a decomposition method, to clustering, like, or decompose the mediator matrix? Because I feel the pathway interpretation is very nice, but why, from the very beginning, why you choose this method?
Yeah, so in the paper, we propose an ensemble of different methods, and I didn't have time to go through every single one of those options. So, you know, we applied sparse principal components analysis, we applied the BAMA method as well as pathway lasso. I think why I wanted to present the population value decomposition approach here is, first of all, it is unique in that it incorporates this structural equation model that borrows information from the exposure matrix and the outcome variable. The second thing is that it fits very nicely when you have a biological hypothesis. Like, you know, there's a grouping structure ahead of time, and you're going into this analysis with a strong hypothesis. And we believe that we had a strong hypothesis at the onset of the study. And I tried to illustrate that to you with that heat map. We've explored through several different studies building up to this, that there's ample biological evidence here. So those are our justifications.
Yeah, this is quite interesting because I work in metabolomics world, so we're dealing with these metabolic pathways a lot and try to interpret whether the pathway itself actually has any mediation effects or like a causal effect. But one thing is that I just wondering, because individual metabolites can participate in different pathways. And a lot of time when we saw multiple pathways, but these multiple pathways can share common metabolites. And so whether those decomposition method consider this kind of, it's not totally orthogonal to each other, basically, but the single metabolites can contribute to multiple pathways, whether that be considered into this framework of method, and whether we can still use that method to interpret that.
Yeah, I think you present an important point that I've had really great discussions with other folks too on similar problems. In the current framework, our analytical proposals does not incorporate for that particular problem, but I think that's certainly another situation that we need to think about in the future. So it's a really exciting thing, and I'm particularly excited about thinking of ways to do that. Especially, there's also this problem where, you know, some of these metabolites might be affecting the concentration of another pathway, right? And so there's this like layered approach too, and I'm excited to explore this going forward. But in this particular framework, it's assuming that there's strong boundaries between the different groupings.
Right. Is it possible that you can share us this kind of ensemble paper that you test out all these methods, like different methods, and we can learn more? And I hope to keep contact with you.
Absolutely.
Learn from you.
Yeah.
Thank you.
That's a Nature communication paper, and I think that I really appreciate that they had we had to deposit, like, you know, template data, all organized code for all the methods in order for their GitHub and our GitHub as well. So we have that ready, annotated code. So I think that's one thing. I think that is this grouping structure. So our problem really, if we apply BAMA, or that's why I mentioned this in future research, in the variable selection methods, there is not an immediate way to incorporate the grouping structure as of yet, right? Because I'm just tossing everything similarly, I'm not taking that into account. So how to really think about this co-selection of correlated mediators, and also extend maybe mediator selected, the targeted penalization that we did, which has an a priori group structure.
Yeah.
So something like a group selection of targeted that, and with overlapping groups, that could be a very nice extension of the variable selection work that has been done in this domain, but transfer it to natural indirect effects.
Yeah, I agree. Thank you.
Bhramar, in the 4-component mixture, multivariate mixture model that you described at the end, you seem to have reverted back to something that sort of looks like a spike and slab, in that 4th component is a 0, 0 exactly, and the null components for the 2nd and 3rd component of the mixtures are also have 0 variance. You want to comment on why you did that rather than continue with the other rest?
That's a great question. I was actually thinking that you might ask me that. So we did try to put like very small variances in those components as well. But there were some identifiability issues when you go to 2 dimensions. It was very hard to actually have two. So basically we have the first variance component, the first component, we have a full matrix. The second component, we have 1, 0, and other 1 non-null, right? So in that, but it was very hard to tease apart when we started with 1 large, 1 small, 1 small, 1 large, and 2 small. That framework, it was very hard to pin it down. So when we went to that formulation, so you'll see that even our second and third component also has 0, right? For the variance, it's at only 1 non-null component.
Even if you constrain the null component variances to be the same in the second and fourth, third and fourth?
Yes, so I think that is those are some of the constraints probably we needed to put in that we did not. So if we made the same, probably this will work better. But with all free choices, we ran into problems.
Well, it doesn't surprise me. It's expecting a lot of the...
Yeah, but that's a great idea that you can actually set them to be same. Mm-hmm.
Other questions for either of them? Well, thanks again, both of you. And thanks.
Guys,
thank you for having us here.
We have so much in common, so let's just keep up this dialogue. Thank you.
No, thank you so much.
Thank you so much.
Thanks, everyone. Bye-bye.
Bye-bye.
Bye.